This is a signal pathway of HGH, or human growth hormone. It all starts in the brain as the hypothalamus sends a signal to the body that needs to grow. From the hypothalamus, that signal gets carried down the pituitary gland to make the human growth hormone. This hormone does not make the cells grow, it sends a signal to kickstart the process of muscle, bone, and tissue growth. The pituitary gland secretes HGH as it travels to the bloodstream via the endocrine system to the liver where the receptor awaits. This is the start of the signal transduction pathway as the HGH ligands bind to the HGHR, or the human growth hormone receptor, extracellularly. The HGHR is a membrane-bound receptor. As it's embedded in the phospholipid bilayer, it forms a complex with tyrosine kinase, which activates the ligate binding. The HGHR complex binds with the ligate, which creates a conformational change. The Janus kinase, or JAK2, goes to being parallel crossing over itself, activating STAT3 by phosphorylation. The signal transducer and activator of transcription, or STAT, travels into the nucleus to transcribe various genes. This is how the insulin-like growth factor, or IGF-1, is then made to be secreted outside and travel to the bones and tissues to signal to start mitosis. This is a cell and a bone interface. A cell spends 95% of its time here growing, replicating DNA, and preparing to go through mitosis. This cell just passed the G2 checkpoint, meaning the cell has enough DNA space, centrioles, and nutrients to undergo mitosis. The first stage is prophase. The microtubules and associated proteins start to assemble and prepare to attach to chromosomes. The chromatids start to condense into chromosomes and pair off into sister chromatids. They're closely attached to the centromere. The nuclear envelope dissolves. The second stage is prometaphase. This is where the microtubules attach to the chromosomes at the kinetochore and begin to move towards the center of the cell. The third stage is metaphase. This is where the centrioles line up the chromosomes in the middle of the cell and prepare to be separated. This is where the M checkpoint occurs, as the checkpoint makes sure the chromosomes are correctly aligned in the center of the cell, as well as attached to the microtubulars correctly at the kinetochore. The fourth stage is anaphase. This is where a signal is sent out from the kinetochores via proteins that walk along the microtubules to start shortening. The non-kinetochore microtubulars elongate as the kinetochore microtubulars shorten, which pull the sister chromatids apart to the opposite poles of the cell. The last stage is telophase and cytokinesis. As the microtubulars disassemble, the chromatids start to turn back into chromatin as the nuclear envelope reforms. The other needed organelles go into the two new daughter cells as the cytoplasm finally separates, creating two identical daughter cells. The bone tissue has started the process of elongating to create a larger human being.